So tonight's video is going to be about translation. So we're going to finish up the process of protein synthesis, and then we'll go ahead and model this one and stuff in class as well. So translation is going to occur on the ribosomes. Remember that translation is taking that mRNA and converting it into a protein or into a polypeptide. And so both prokaryotic and eukaryotic cells are going to do translation, just like both of them did transcription. Remember, this is a fundamental part of being alive. You've got to make proteins. So both prokaryotes and eukaryotes do both transcription and translation. And both of them have ribosomes. Remember, ribosomes are not a membrane-bound organelle. So all prokaryotes have them as well as eukaryotes. And the ribosomes can be considered what is called free. You see this one right here, a free ribosome or a bound ribosome. So these right here are considered bound ribosomes. So bound ribosomes are found free-floating in the cytoplasm, and bound ribosomes are attached to the endoplasmic reticulum. That's actually what makes the rough endoplasmic reticulum is the attachment of these ribosomes. So prokaryotic cells will only have free ribosomes because they don't have an endoplasmic reticulum, where eukaryotic cells will have both free ribosomes in the cytoplasm as well as ribosomes attached to the rough ER. So the ribosome's job is to help the tRNA, the anticodon on the tRNA bind to the codon on the messenger RNA. If y'all remember with tRNA, it's shaped kind of like this, kind of like a clover. And one side has the anticodon, which is going to have like a ACG or GCA. It's going to have 3 nitrogen base letters there. And the other side is carrying the amino acid attached to it. And so this side right here with the anticodon is how it binds to the messenger RNA. For example, let's say our mRNA was ACG. So the anticodon that would be on the tRNA would be UGC. And so that we'd have our tRNA there, and it would bring over a particular amino acid. And so the ribosome's job is to help this anticodon and codon attach to each other so that the amino acid can get dropped off and attached to form the polypeptide or the protein. So ribosomes are made out of rRNA, or ribosomal RNA, and proteins, and they have 2 subunits. They have a large subunit and a small subunit. And when the ribosome is not actively doing translation, those 2 subunits are found separately. When messenger RNA arrives, they will actually come together. You can see the large subunit here, and the small one is this lighter purple. And then when the messenger RNA arrives, they form into one larger, fully functioning ribosome. So first we're going to look at translation in a prokaryote. So translation in a prokaryote is going to look a little bit different than translation in a eukaryote. Because if you remember, for a prokaryote, right, I don't have my DNA in a nucleus. So transcription and translation are both going to happen in the cytoplasm because the DNA is found out in the cytoplasm of the prokaryotic cell because there is no membrane-bound organelles. So when transcription and translation are happening in the prokaryotic cell or in the bacteria, remember all prokaryotic cells are bacteria, happening in the bacteria cell, they're going to be happening simultaneously, or they'll be happening at the same time. So a couple of things that would be different. In addition to the fact that both transcription and translation are happening out in the cytoplasm, when that messenger RNA is created, there's not going to be any messenger RNA editing. In the eukaryotic cell, in our last video, we talked about how we got a poly A tail added and a 5' cap was added, and we spliced out the introns to then put the exons back together. None of that messenger RNA processing happens in a prokaryotic cell. And the ribosomes are reading that messenger RNA as it's being transcribed. These 2 things, like I said, are happening at the same time. I think it's easier if you look at this picture over here on the right. So my blue is my DNA here. So I have this strand of DNA. The RNA polymerase is traveling along that strand of DNA, so it's making messenger RNA. That's the pink strip that's coming out there, it's making that messenger RNA. So those ribosomes are grabbing a hold of that messenger RNA basically as soon as it's being made. And so they are making proteins at the same time that the RNA polymerase is traveling down the strip of DNA and making the messenger RNA. They will continue to, the messenger RNA basically will get fed right through these ribosomes, and it will be continuously they will continuously be spitting out different polypeptides or proteins. So in a eukaryotic cell, translation is going to happen separately from transcription, and it's going to involve a series of steps that are going to require energy. So it will require energy to do translation, and it's going to have three basic steps: the initiation, elongation, and termination. And we're going to talk about each one separately. So we're going to obviously start with initiation. So initiation is going to be the start or the beginning. So some things that need to happen during the initiation process. The rRNA that's in the ribosome needs to first interact with the messenger RNA. So what that will do is that will allow for step B here. When that rRNA in the ribosome interacts with the messenger RNA, that will cause those two, remember we had the big large subunit and then the smaller subunit. So when that messenger RNA comes in there and they interact, that will allow these two ribosome subunits to come together to make the complete, fully functioning ribosome. So as the messenger RNA moves through the ribosome, remember it's read in groups of 3. Remember those groups of 3 are my codons, right? If I'm looking at this messenger RNA in my picture down below, right, break it into my groups of 3. And those groups of 3 are going to be my codons, 3 nitrogen bases. So one codon is going to equal 1 amino acid. So we can see if we follow all the way down to the polypeptide chain, this one codon here, AUG, so those 3 nitrogen bases code for 1 amino acid, methionine. So if I had a messenger RNA sequence that had 300 codons on it, then it would code for 300 amino acids, because one codon codes for 1 amino acid. But if I had a RNA strand that had 300 nitrogen bases, well, they're done in groups of 3, and so that would code for 100 amino acids. So one codon, 1 amino acid, one codon is 3 nitrogen bases. Okay. All of your messenger RNA is going to have a start codon, and that start codon is always going to be this AUG, and that will code for methionine. So every messenger RNA is going to have that start codon. That start codon, when the ribosomal RNA, so when the rRNA interacts or binds with that start codon that's on the messenger RNA, that is initiation. That's officially when initiation is going to happen. So the rRNA binds with the start codon on the mRNA. That makes the ribosome come together into a fully functioning unit, and that's the initiation process. So those codons are going to be used to code for the specific amino acids. And if you remember this from your freshman year, right, we use our codon chart to determine which amino acid they are coding for. Okay? So the coding process, the coding itself is considered redundant. And by redundant, we mean that we have multiple codons for every amino acid. If you look at the chart, I have UUU and UUC both code for phenylalanine. I have 4 right here that are going to code for serine. So the code is considered redundant since I have multiple codons that can code for each amino acid. The start codon is always going to be AUG, which will translate into methionine. And we have 3 different stop codons. And those stop codons, those are what are going to make us stop translating the mRNA sequence. They're going to end the polypeptide chain or the protein. And so this code is the same for all life. So if I have a CUU codon in a bacterial cell and that codes for leucine, that codes for leucine for us. It codes for leucine in mushrooms. It codes for leucine in trees. It codes for leucine in algae, in any organism. So that code is common. Your freshman year, we called it the universal genetic code. So it's code for all, it's the same code for all walks of life. So that's one of our strongest supports for a common origin for life. And so we'll revisit this some when we talk about evolution and we talk about these common origins and these things that provide support for this theory of the theory of evolution. So just a quick reminder, if you don't remember how to read your chart, remember the first space is always over here on the left. So we go in whatever row, like let's say our codon was, I erase this arrow here. Let's say our codon was U, G, U. So our first codon, our first nitrogen base here would be U, so I'm up here in this U row. Okay, my second one is always along the top, so my second one is G, so I'm over here, which means now I'm narrowed down to this little box right here. And then I'm going to use the U, so there's multiple U's over here on this far right side. I'm going to use the U that corresponds to the appropriate box that I'm in now. So I know that I'm at cysteine here. So we'll practice with the codon chart, but it is exactly the same as it was your freshman year. So these codons are what are going to dictate which amino acids are attached. And so this is going to be our process here of elongation. The tRNA, or the transfer RNA, remember tRNA is transfer RNA. The transfer RNA is going to match the amino acids based upon those messenger RNA codons. So what will happen with the tRNA? So here's our tRNA in your this picture down here on the left. So I've got my anticodon on one side, and I'm going to have my amino acid attaching on the other side. This particular tRNA, whose anticodon is AAG, the amino acid that is on this other side, it never changes. It's always the same amino acid. For example, AAG, if that's what's on my tRNA, that would go with UUC on my messenger RNA. So when I read my codon chart, it's based off of my codon, right, on my messenger RNA. So when I read my codon chart, UUC is serine. No, I'm sorry, it's phenylalanine. So phenylalanine is this amino acid that's attached on this end of this tRNA. So that tRNA cannot pick up any other amino acid. So if it carries that phenylalanine over and drops it off at the ribosome, when it leaves, it will go and pick up another phenylalanine. That's how the cells and stuff know that it's going to put the right amino acids in the right order. These amino acids that the tRNAs carry are specific. They cannot change. Okay? That tRNA cannot go pick up whatever amino acid it wants to. Because remember, tRNAs are recycled. They basically drop the amino acid off, and then they'll go back and they'll pick up another one. But again, that amino acid is going to be exactly the same. They don't go back and pick up a different one. So the tRNA is going to match the amino acid that it's bringing, it's going to pair up based on the mRNA codons. And the amino acid that it carries is consistent. So the new amino acid is going to be added to the growing polypeptide chain. So this is the elongation part. Right? We had the initiation part where we started it, the elongation part where we made it a little bit longer, and then we're going to have the termination part. So you can see over here on the right, you can see in the ribosomes, these kind of weird shaped things here, those are the tRNAs. You can see their anticodons on the bottom. You can see how they have amino acids, these purple things attached on the top. And so you'll see how they come in. This tRNA with this glycine on it is entering the ribosome here into spot number 1 here. So it's entering the ribosome there. It'll get shifted down to the second spot. When it gets shifted down to the second spot, it'll attach that glycine onto that phenylalanine, and then they'll get kicked out to go pick up another one. Then when they're empty and they've dropped off their amino acid, they'll get kicked out to go pick out another one. So this is how our elongation process will work. These remember, the ribosome's job is to help the anticodons and the codons bind together so that then the peptide bond can be formed between the amino acids that are being brought in. So this process will continue until the stop codon is reached. And once the stop codon is reached, that's when we've hit termination. So the process will terminate or will stop by releasing the newly made polypeptide. So remember, the polypeptide is the protein, right? So this newly made polypeptide will be released off into the cytoplasm so that it can either do its job in the cell or it can get packaged and shipped out, just kind of depending upon whatever its job it is. Depending upon the messenger RNA, it may get translated again. The ribosome will split back apart into its two subunits until it needs to be used again. But the process will stop, and that protein now will be available for use. So the exception to this process would be what are called retroviruses. So retroviruses have what is considered an alternate flow of information. By that, we mean that instead of the information going DNA, because most of our information flows DNA to RNA to protein. With a retrovirus, it goes from RNA to DNA, and then it will eventually end up back to a protein. It'll go back to RNA and a protein. Okay, so how this will work is these retroviruses will use, an example of this would be HIV. HIV is the most common retrovirus example that you'll see. So they'll use an enzyme here called reverse transcriptase. So I know it's an enzyme because it ends in ase. So this reverse transcriptase does transcription in reverse. It takes RNA and turns it into DNA. So what happens with these viruses? So you can see this virus out here, right? Remember, viruses only consist of genetic material, which could be RNA or DNA, and that protein coat, the capsid protein coat. So the virus and a retrovirus, its genetic material is RNA. So a retrovirus is made up of RNA genetic material and then a surrounding protein capsid. So when this virus infects this host cell here, it releases, remember, it injects its genetic material into the host cell. And so this genetic material is going to be, made of RNA. So if you remember back to your freshman year, right, the virus injects its DNA into the host cell and then uses the host cell's materials to help manufacture its proteins. Okay, well, in this case, the virus is injecting RNA into the host cell. Well, the host cell can't use RNA to do everything that it needs to do. The RNA that came out of the virus needs to be converted into DNA first. So you see the red is the RNA that got injected here first. And so what will happen is this reverse transcriptase will turn that into DNA. So it will make DNA out of that rRNA. And so now there's DNA that will get incorporated into the host cell DNA. So that DNA that was formed using that reverse transcriptase gets incorporated into the host cell DNA. And now the host cell is going to go through its normal processes to make proteins for this virus. It will do, when it transcribes its own DNA, it will transcribe that viral DNA. And now that viral DNA that new viral RNA can go through and it can do translation and it can make the proteins for the new virus. So we'll do some more work with both of these things in class. We'll fully model translation again in class, and we'll talk a little bit more about retroviruses in class. But this is important at this stage because we need to realize it's basically an exception to our DNA RNA to a protein rule.